When we introduce the topic of movement, we have to distance ourselves from some fond dreams we might have of the brain as a controller. That is not a helpful way of understanding the role of the brain. And as we've studied movement, we've found little to support overly psychological interpretations of the brain. But it's time now to return and consider the role, what we know about the role of the central nervous system or the brain in movement. And this is not going to be a simple story. We know that for coordinated movement to happen, we need an appropriate environment. Remember the role of the floor in walking, or the self-organizing gait of the decerebrate cat. So when we look at the nervous system, we can recognize that different parts of the nervous system are involved in the production of coordinated movement in different ways. And we're going to look at three areas in particular. We're going to look at the primary motor cortex, the cerebellum, and the basal ganglia. In each case, we're not going to say this part of the brain is for X, Y, or Z. That kind of speak is usually unjustified. What we are going to do is recognize the involvement of these parts of the brain in certain recognizable characteristics of movement. So we're going to begin with the primary motor cortex. That's shown there in blue, and that's the first gyrus of the frontal lobe here towards the front of the head. And you may remember when we studied perception, the gyrus just behind it, they're called the postcentral gyrus. That is also the primary somatosensory cortex. That's where we can map the touch receptors distributed all over the skin onto the part of the brain and see how sensitive the different parts of the body are. The precentral gyrus, or the primary motor cortex, lies just beside that. And when we perform a similar mapping, as we can do, lo and behold, we find something quite astonishing. In the primary somatosensory cortex, we came to this mapping by looking at where touch sensitivity is on the exterior of the body. If I touch you here, we can register a change there in primary somatosensory cortex. And we got this homunculus you see there on the right-hand side with an exaggerated amount of brain real estate given over to the lips and the face and the hands, all of which are very sensitive, and very little given over to the elbow or the heel, which are frankly not very sensitive. Now, when it comes to the primary motor cortex, we have to build that map a different way. In the primary motor cortex, we find particular cells, the pyramidal cells, which project down through the brainstem and into the spinal cord. They're very, very long, these nerves. Some of them can be as long as this, a single nerve as long as this, tiny, or very thin, but very long. And there they have a synapse and they project to nerves that go out to the periphery, to the arms and legs. So we can follow those paths and we can see where do these primary motor cortex, the cells in primary motor cortex project to, and we can reconstruct a homunculus. And it's the same guy. In fact, if we reconstruct them a little bit more visually, there's the sensory homunculus on the left and the motor homunculus on the right. It's the same guy. Why is the same guy hiding in two different places on the brain? One of them ostensibly to do with perception, one of them ostensibly to do with motor control. Well, the answer is our psychological theories of the brain are not really well supported by the brain's organization itself, but the brain organization itself can tell us an awful lot. What it can tell us is that the sensory part of our being is not separate from our motive being, that perception and action, which psychologists very often like to separate as input on the one hand and output on the other, that's not really how the brain is organized at all. So that's kind of interesting. Then at the back of the brain, we find this little organ. It sits beneath the cerebral cortex. It's much older from an evolutionary point of view. And it's structured almost like a second little brain. The cerebellum has two hemispheres, which resemble closely the neocortex, which sits above it. We said we're not going to say what a part of the brain is for, but we do want to understand how these various parts of the brain are involved in the production of coordinated movement. We know this mainly from pathology. We do know that if you damage your cerebellum, there are typical movement problems that arise. And this gentleman, who's an expert on patients with those kinds of problems, will now demonstrate to you just the kind of disturbance to movement that happens when you have cerebellar disease.
The last gait that I'm going to demonstrate is the ataxic gait. In the ataxic gait, the patient has difficulty narrowing the station and maintaining balance. So typically they'll have a wide stance, is trying to maintain balance, and that there oftentimes will be unsteadiness in the trunk. So there may be some truncal titubation, which is an anterior-posterior type of tremor at about 3 hertz. And there's also a tendency to lunge or to jerk sideways, and the patient then has to catch themselves. Of course, one of the ways to bring out this particular type of problem is to narrow the station, asking the patient to walk tandem. So if I then am ataxic, I then try to walk tandem, then I'm going to have difficulty walking.
Okay. Before we move on to the basal ganglia, the cerebellum, when it's damaged, it produces this kind of disordered gait. With that, we can say it plays a significant role in stabilizing patterns. And through lots of other experiences we've had with the cerebellum, we can say that when you learn a pattern first, maybe you're learning to play the piano or to juggle or something like that, it's a bit uncoordinated. As you get more expert at doing something, it becomes tighter and more coordinated, highly controlled, well, very tight, shall we say, in its organization. The cerebellum plays a special role in keeping that tight coordination and time. Thirdly, we're going to look here at a whole bunch of structures. You can see loads of them there, and they're very varied, and they've got lots of different parts to them, and we don't understand them very well at all. But these are deep within the brain. They're not part of the neocortex that we've seen so much of. From an evolutionary point of view, these are much older than the neocortex. They're found in all mammals, but they're found as far back as the last common ancestor of the human and the frog. So these are very old structures indeed, and they're totally highly differentiated. People love to tell stories about what parts of the brain are for, so there's all kinds of fanciful stories out there. When these break down, there are again characteristic symptoms and effects. And it's unwise to say what a part of the brain is for, but based on observation of disturbances that happen when the basal ganglia are damaged, we can recognize that they're playing a role here, something like the conductor in an orchestra. Now think of the conductor in an orchestra. The conductor has a whole lot of musicians at their disposal. The conductor doesn't have to know how to play the oboe. The conductor doesn't have to know how to play any instrument. But the conductor will play a central role in sequencing, in telling things when to start and when to stop, and in regulating intensity and saying, bring up the strings, tone down the percussion, bringing things in and out and modulating intensity. That seems to be the role of the basal ganglia considered as a whole, but there's all kinds of detail that we haven't gone into there. So those are the 3 parts of the nervous system where we have some insight into their role in the production of coordinated movement. The primary motor cortex is the primary source of nerve impulses to muscles. The cerebellum, which has something to do with stability, posture, and the control of well-learned coordinations. And the basal ganglia, which have to do with the sequencing and fine-tuning of sequences of skilled actions. So we've looked at the areas of the central nervous system that seem to be involved in the kind of movement we've looked at throughout the animal kingdom, which has mainly been locomotion. And we've concerned ourselves, because you have to start somewhere, with movements that have been largely cyclic, repetitive movements. But not all movements are of that nature. Locomotion is roughly periodic, And periodic movement we understand quite well now as a matter of bringing oscillating components to bear on one another. But humans in particular produce very many actions which occur in sequence but without repetition. One of the domains we do this is speaking. In speaking, it's not simply repetitive ba 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 ba. You couldn't communicate if you were going to repeat yourself like that. So we have complex sequences of movements which are very sensitively timed and which are at the service of the demands of language. So speech introduces all kinds of questions that we've met nothing that can help us here. First of all, when we consider movement and speech, we realize that movement happens at 3 different timescales in speech. On the one hand, there's the relatively slow movement of the diaphragm and respiration, which is, of course, essential to speech because speech is controlled exhalation. And that is, you take a breath every couple of seconds. Then we've got the movement of the articulators, the tongue, the mouth, the lips, the teeth, and they're moving at about 5 cycles a second. We produce in speech about 5 syllables per second, an amazing number, and each syllable has its parts as well. And then there's the vocal folds in here, which are producing the buzzing noise of voicing, and that moves somewhere between 80 to 200 or 300 times a second. So radically different timescales, and all this has to work together. Another weird feature of speech is that all those moving parts are in relative isolation from the outside environment. When we consider most kinds of movement, we have to consider the environment in which they happen. You won't understand walking unless you understand that there's a floor that's essentially involved. You won't understand even how you use a pencil unless you consider the structure of the pencil. But all the speech movements happen back here in relative isolation from the outside environment. We've got this vast range of timescales, and we've got this marshalling in the services of language production. So speech shows us just how ridiculously complex this can get. One thing that we notice is, you remember on the homunculi, the somatosensory homunculus and the motor homunculus, both the orofacial region and the hands had large amounts of real estate. Well, it's no big surprise that we find movement of the hands to be integrally related to movement of the mouth. The same areas of the brain which, if damaged, produce changes in speech articulation, often also produce changes in the ability to finely control the fingers. So there's a deep link between our voices, our speaking, and our use of the hands, which is poorly understood. So we'll wrap it up there, except to note that when it comes to studying movement, we cannot kid ourselves. We have to be driven by the facts, driven by our commonality with animals, driven by the physics of the matter. And it's here that many of our fantasies about the brain sort of reach a low point. The brain actually doesn't move very well, as this horror film illustrates. It's my favorite brain. Beware of brains.